Good morning. Myself, Dr. Anil Kumar Chaudhary, in charge Department of Genetics and Plant Breeding, College of Agriculture Science, TNU, Moradabad. Now, today we discuss on the crop improvement second, BAG 611. Today topic is plant breeders kit. About the overall introduction of this subject. So first of all, what is the plant breeder kit and what is the usage of plant breeder kits? So first of all, I tell you about the aim of the experiment. To study about the plant breeders kit and its uses. Introduction. The plant breeder frequently use the different tools, instruments and materials to carry out the selfing, artificial crossing and emasculation, pollination, selfing, tagging, bagging and hybridization process. So taking the observations. So first of all, we have need to magnifying glass or lens. This is the small or medium size glass handle, and purpose of this magnifying glass to observe the anthers. Anther is the mature, or anther is observe the small flowers, buds, stigma, and other parts of the flower. That is the major use of the magnifying glass. And second instrument we use the breeder kit forceps. We can use the forceps medium size or long size, but the forceps should be the pointed. And purpose of the forceps for emasculation and removing the anthers from a flower. And it is the mandatory to all breeders. Flower should be proper mature, not over mature, not premature. Because in premature flower, small anther size is very small, and after the over mature of the anthers, anther can be blast. So we should need remove the anther from a stigma bud through the forceps on time. And next instrument and brush. The brush after the emasculation, after the removal of anther in a bud, we need within time, according to the species, according to the crops, we use the brush for the collection of the anthers from male part of the flower and to carry with the help of brush. So we can use the brush size 3 or 4 point for the collection of pollens and placing the stigma to surface. It is very useful when little quantity of the pollens are used. And next is bag. After the emasculation and pollination, we have very need the bags because the anther enter in the ovary within 1 hour or maximum time. So protect the female part with the help of different type of the bags like muslin paper bags, cotton bags and plastic bags. Depend on the crop and species and size of the bags. So we use this type of the bags, different type of the bags, papers, brown papers bag, muslin cloth bags for covering the flower or bud, entire inflorescence, yeah, whole plant. This bag can use for selfing. If we want to produce the progeny, plant progeny, pure progeny, to determine the contamination, to we use the whole plant bag. So that is the very most important, and we can use the bag proper um, bindings and ethyl alcohol, smile oil and tags. After the emasculation, pollination and fertilization, all process complete has been, then we need the tags. We write the identification of plants, this plant is male, we use this the pollination, yeah, pollens from this plant. So we write on the tag this type of the uh, plants. And there are three types of the tag: one dwelling tag, the second plant tag, third silver or aluminum foil tag. Depend on the plant, depend on the crossing program, we can use the labeling and mark the identify after collection of the seed. So that is the very most important part. Next, marker, pencil. We use the permanent marker or pencil for writing on tag. In case we use the pen, wall pen and other type of the pen, so in sunlight after 4 or 5 days in rainy season, so writing is invisible. So pencil writing or permanent marker writing is permanent, because the crop complete the duration three in 3 or 4 months. So that is very essential part. Next, meter or scale, measuring tape for the measurement. Like in mustard, if you want to take the data like plant height, number of primary branches, number of secondary branches, number of silicua, length of main shoot length, and length of branches. So different type, and measuring the plot size, different type of the use of measuring tapes or scale, or we use the scale in this such type of the experiment, and take the data in centimeter or millimeter upon the situation. And next, wing balance for 1,000 seed weight, for plant weight, leaf weight, branch weight, and fruit weight, product weight. Such type of the weight used in experiment. So we have very essential item. And the field book. Starting the experiment, first of all we have need the field book, because we have mentioned a lot of things like our what is our experiment plan, layout, and we have genotypes, where is the site of genotypes, where is the uh, location, and what is the sowing date, which type of the crossing made between the different type of the genotypes, all things notes in the field book. And after the harvesting, what is the current status of the F1 progeny, F2 progeny, any type of the progeny we mention in field book. And take the data, plant data, number of balls, number of silicua, number of pods, weight of pods, number of seeds, plant height, different type of the data based on the crops. We mention all things in field book every year and compare. So that is the essential. And next, needle for open the bud. In case we want our pollen is mature or not mature, over mature, premature, what is the situation of the pollen grains, what is the situation of the anthers, what is the situation of male female parts. So first of all we open the bud with the help of needle. So that is very essential item. And the needle is long with plastic handle, that is mandatory. So thank you. This is the small introduction about the plant breeders kit, and used in the crop improvement plant breeding program. So that is the main things in plant breeding department. Without plant breeder kit, we cannot complete any type of the experiment. And Last year we sown the 8 genotypes of Indian mustard and wheat crop. So crossing between the NRCHV 101 and NRC DRC2, RS ASO 25, RS ASO 49, made a half daily between the 10 genotypes. So we use lot of time of the plant breeder kits. So thank you.